Welcome back to Bookfulness. This is your deep dive, where we really get into the fascinating sources and guide you through the knowledge inside.
That's right.
Today, we're continuing our intensive look at this landmark text, Evolution.
A foundational work, for sure.
Okay, let's unpack this. Last time, we officially wrapped up Unit I. We explored those core concepts, didn't we? Natural selection, adaptation.
We talked quite a bit about how the environment acts, well, like a sieve.
Yeah, sorting out which traits survive and reproduce.
Yeah, and it's an incredibly powerful sieve, absolutely. But, it only works on what's actually there.
Right.
It can only sort the deck of cards that's been dealt, so to speak. So as we move into Unit 2 now, how evolution works, we're shifting focus
From the sorting process.
to the source material itself. Where do those cards come from in the first place?
Exactly. And that's our mission today. We are exploring the ultimate, the fundamental source of all biological diversity, variation and mutation.
Crucial stuff.
Yeah, if you think of evolution like building a house, mutation is kind of like getting brand new specialized tools delivered.
Okay, I like that analogy.
And variation, that's the pile of lumber and bricks you already have on site. That's what selection gets to work with.
Right. And without that initial variation, that pile of materials,
The building just stops, cold. No variation, no evolution.
it sounds almost too simple, but the core idea from this chapter, the thing you really need to internalize, is just that. Variation is the fuel.
The absolute starting point.
Yeah. If every single organism in a population were genetically identical clones, basically.
Then natural selection has nothing to choose between.
Exactly. Nothing to select, and therefore no evolution could possibly occur. It would just stall.
And we need to be super clear about what kind of variation we're talking about here.
Yeah.
Because selection, well, it sees the physical differences, right? The color, the size, maybe the speed.
What biologists call the phenotype, the observable traits.
Right. But for evolution to actually happen over time, those differences, those phenotypes, they have to be inheritable. They need to be passed on.
Precisely. For selection to drive change across generations, the differences we see, the phenotype, must have a basis in the underlying genetic information, the genotype.
The DNA sequence.
That's it. If, say, a giraffe stretches its neck really hard during its lifetime.
The old Lamarckian idea.
Right. If it does that, but its offspring don't inherit some sort of gene for a slightly longer neck.
Then nothing changes for the species.
Correct. Selection is just acting on that one individual's life history. It's not evolution of the species. The change has to be rooted in the genotype.
Okay, so let's quickly revisit the machinery that makes this inheritance possible, starting with the big one, DNA.
The master molecule.
It's incredibly long, built from those 4 famous chemical bases. You know, the letters A, T, C, and G.
And the pairing rule is strict. A always pairs with T, and C always pairs with G. That's the foundation of the double helix structure.
And the scale here,
Yeah.
just mind-boggling.
It really is. I think that's something people often don't fully grasp. This isn't just like a short instruction manual. It's an epic.
Yeah.
The average human chromosome, we're talking well over 100 million base pairs, just one chromosome.
Right.
And the entire human genome, all your chromosomes together, that's estimated at about 3.2 billion base pairs.
Billion. With a B.
That is a staggering amount of information packed into the nucleus of almost every single cell in your body.
I read somewhere if you printed out the human genome, just the letters A, T, C, G, it would fill something like 500 standard textbooks.
That sounds about right. It's a library, truly, in every cell.
Okay, but here's where it gets weird, right? The text points this out.
Yeah, this is one of the most counterintuitive things in biology, I think. That scale check, the size of the genome,
Yeah.
it doesn't really correlate with what we think of as complexity.
You'd think it would. You know, humans, elephants, dolphins. Big brains, complex behaviors. Surely we'd have the biggest instruction manuals.
You'd think so, but nope. There's surprisingly little correlation between an organism's complexity, however you want to measure that, which is tricky in itself,
Sure.
and the sheer size of its genome. We find, certain plants, some single-celled organisms, protozoa, even some salamanders.
Salamander.
Yeah, they can have genomes that are hundreds of times larger than ours. Our 3.2 billion base pairs. It's actually kind of middle of the road in the grand scheme of life.
So if complexity isn't the driver, what are researchers thinking? I mean, all that extra DNA in, say, a huge plant genome, what's it doing?
Yeah, that's kind of the million-dollar question, isn't it?
Why carry it all around? Seems like baggage.
Well, it points to something fundamental. Evolution isn't always neat and tidy. It's not always perfectly efficient.
Okay.
Some of it's probably just beat, honestly.
Mm.
Segments that built up from replication errors over millions of years. And selection maybe hasn't gotten rid of them because the cost of carrying them is, well, pretty small, marginal.
Right. Doesn't hurt enough to be actively selected against.
Exactly. But a lot of it, even the stuff that doesn't code for proteins, likely plays some kind of role. Think regulation turning genes on and off.
Ah, okay.
The control systems.
Or, structural integrity of the chromosome itself. Maybe it helps with packaging the DNA correctly.
I see.
So it might be less about what the organism is in terms of complexity and more about, say, how easily the genome can be reorganized or how stable it needs to be over time.
Oh.
So yeah, genome size could just be like the result of this ongoing race between errors piling up and how good the organism's machinery is at cleaning those errors out, like DNA repair mechanisms.
Fascinating. Okay, so wrapping up the basic architecture. These huge strings of DNA, they're organized into structures called chromosomes.
Right. And in eukaryotes, like us, plants, animals, fungi, we typically have pairs of each chromosome were deployed.
2 copies.
2 copies.
Yeah.
Whereas prokaryotes, bacteria, and archaea, they're usually haploid, just 1 copy.
And within those chromosomes are the functional units we call genes. Yep. Genes are the specific segments that, do something. Typically, they carry the instructions, the code for building proteins.
And proteins do almost everything right. Build tissues, act as enzymes.
They're the workhorses of the cell, absolutely. And humans, it turns out, have roughly 20,000 protein-coding genes.
Which sounds like a lot, but given our complexity, maybe not as many as you'd guess.
Yeah.
Especially compared to that huge genome size.
Exactly. It highlights how much of the genome is not directly coding for proteins. But, okay, let's get to the code itself. How is that information read?
It's read in blocks of 3. 3 bases make a codon.
That's the key. And each codon specifies a particular amino acid, which are the building blocks of proteins, or sometimes it signals stop reading.
And the text makes a big deal about this triplet code. It's basically universal.
Almost entirely universal, which is just astounding when you think about it. From bacteria living in volcanic vents to us, to redwood trees, we all use essentially the same dictionary to translate DNA into protein.
And that universality is huge evidence
for evolution, for a single common origin of all life we know. If life popped up independently multiple times, or if the code changed drastically after major groups split off,
you'd expect different genetic dictionary.
You absolutely would. The fact that, say, AGG means the amino acid arginine for you, for a mushroom, for that E. coli in your gut, it points straight back to a shared ancestor deep in the past.
Okay, but here's a subtlety. There are 64 possible codons, right? 4 bases taken 3 at a time, 4 by 4 by 4, but only 20 common amino acids.
Exactly. So the code is redundant. There's more than one way to spell most amino acids.
And that redundancy, that's where evolution gets some wiggle room.
Precisely. This leads us to define 2 crucial types of point mutations changes to a single base pair. First, you have synonymous mutations.
Silent mutations, right?
This is a single base change, but because of the redundancy, it does not change the amino acid that gets put into the protein.
So the DNA sequence changes, but the final product, the protein, stays exactly the same.
Correct. If you look at the codon table, like in figure 4.2 in the text, you'll see for many amino acids, changing that third base in the codon often makes no difference. GGU, GGC, GGA, GGG, they all code for glycine. A mutation from one to another there is synonymous.
Okay, so that's one type. What's the other?
The other is a non-synonymous mutation, and this one does change the amino acid sequence.
This is where things can really change functionally.
Oh, absolutely. The classic example used in the text with figure 4.3 is the beta hemoglobin gene. A single base change in the codon for the 6th amino acid.
From GAG to GUG.
Exactly. That single change swaps the amino acid glutamic acid for valine, and that one tiny change causes sickle cell anemia, a major phenotypic effect from one base change.
Wow. And the text notes that changes in certain codon positions are more likely to be non-synonymous.
Yes, particularly changes to the second position of a codon. They almost always result in an amino acid change, so they're highly likely to alter the protein structure and therefore its function.
Which usually means making it worse, right? Since proteins are generally pretty well tuned by past evolution.
Usually, yes. Random changes to a finely tuned machine are more likely to break it than improve it. We'll come back to fitness effects later.
Okay. But let's not forget that vast expanse of non-protein coding DNA. What about mutations there?
Right, back to the 98.5% or so of the genome that isn't coding for proteins. We absolutely cannot ignore it. Point mutations in these non-coding regions can still have massive effects on the organism.
How does that work? Give that cake analogy again.
Okay, so think of the protein coding gene as the recipe itself, the ingredients list for the cake.
Right.
The non-coding DNA, especially the regulatory bits near the gene, that's like the instructions for using the recipe. It's the oven temperature dial, the timer setting, maybe even instructions for when to bake the cake versus when to make cookies instead.
Right.
A non-synonymous mutation changes the ingredients in the recipe. Might make the cake taste funny, but a mutation in a non-coding regulatory region. You haven't changed the ingredients at all. You've changed the instructions. Maybe the mutation cranks the oven temp way too high.
So you burn the cake, even with the right ingredients.
Or maybe it breaks the on switch for the oven entirely for that gene in certain cells. Or maybe it tells the cell to bake that cake constantly, even when it's not needed. These changes in gene expression, when, where, how much a gene is turned on, can be just as dramatic, sometimes more so, than changing the protein itself.
So evolution plays with the recipe and the instructions.
Absolutely. It tinkers with everything. And one last structural detail here before we move on. Exons and introns.
Right. Genes aren't just one long, continuous stretch of code in eukaryotes.
No. The coding parts are called exons, and they're often interrupted by non-coding segments called introns.
Which get cut out.
Exactly. Before the gene's message is translated to protein, the cell has this sophisticated machinery that snips out all the intron sequences and splices the exon sequences back together into the final message.
And the cool thing there, which the text mentions, is that sometimes the cell can splice those exons together in different ways, right?
Yes. That's called alternative splicing. It means that one single gene, depending on how its exons are stitched together, can actually produce instructions for several different distinct proteins.
Wow. So that really expands the functional toolkit from those 20,000 genes.
It massively increases the coding potential. It's another layer of complexity and, crucially, another source of existing variation that selection can potentially act upon. Different splicing patterns can lead to different phenotypes.
Okay, so Part 1 laid out the blueprint, the DNA, the code, the basic types of changes like point mutations. Now we're shifting gears.
Right. If mutations create new variations, this part is about how existing variations get shuffled and recombined every generation.
Mixing the deck, essentially. Taking all those different versions of genes that already exist in a population and dealing out new hands.
And this shuffling through processes like sexual reproduction is arguably much more important for driving immediate evolutionary change than mutation itself.
Why is that?
Simply because mutation, especially beneficial mutation, is quite rare on a per gene, per generation basis. But sexual reproduction happens every generation, constantly mixing up the existing genetic combinations. It's relentless.
Okay, so let's get some core vocabulary down first. The text defines locus. What's that?
Locus is basically just a specific location, an address on a chromosome. You could say the locus for the eye color gene is on chromosome 15.
Got it. And at that locus, there could be different versions of the gene.
Exactly. Those different versions are called alleles. So at the eye color locus, you might have the allele for blue eyes or the allele for brown eyes or green, etc. They're all forms of the same gene found at the same address.
And when biologists study a population, they look at how common each allele is.
Yep, that's the allele frequency. It's simply the proportion of all the copies of that gene in the population that are a specific allele. So maybe in a particular human population, the frequency of the blue eye allele is 0.3 or 30%.
And if evolution is happening at that locus,
then that frequency, that 30%, is changing from one generation to the next. It might go up, it might go down. That change is evolution at the genetic level.
okay, let's use the example from the text to see how this mixing happens, starting with segregation.
Right. This is fundamental Mendelian genetics, but let's walk through it. Imagine a really simple population, just two alleles at one locus, AI1 and A2A2.
All right.
And let's start with a weird artificial situation. Exactly half the individuals are pure homozygotes, A0 and A and O-lon. They only have the A dollar in allele.
And the other half?
Pure A2A2 homozygotes. They only have the A2 or 2 allele. So the population is completely polarized. No heterozygotes exist yet.
Got it. So what happens when they reproduce? Assume random mating.
Okay, if they mate randomly, think about the gametes, the sperm and eggs they produce. The A0 and A individuals can only make A dollar in gametes. The A2 individuals only make A dollar gametes.
So the pool of gametes in the population is exactly 50% A01 and 50% A dollar dollars.
Perfect. Now, what kinds of offspring can be made by combining these gametes randomly?
Well, to get an A1A1R offspring, you need an A$1 egg and an A$1 sperm. The chance of getting an A$1 egg is 12. Chance of A$1 sperm is 12.
So multiply those probabilities. 12 times 12 equals
14. So 1/4 of the offspring should be A1A1.
Right. Same logic applies to the $8 offspring. Chance of $8 egg, 12 times chance of $10 sperm, 12 equals 14.
Okay, so that's half the offspring accounted for. What about the other half?
They must be the heterozygotes, A1R tutors, and you can get that combination in two ways.
A$1 egg with A$2 sperm.
Which has a probability of 12 times 12 equals 14.
Or an A$2 egg with an A$ sperm.
Also 12 times 12 equals 14.
So you add those two ways together. 14 plus 14 equals 12.
Exactly. So in just one generation of random mating, starting from that completely polarized population, the offspring generation will have genotype frequencies of 14 A1A1 Oyan egg 1 14 3 1 A2 and 12 A1O2 down.
That's a dramatic shift in the distribution of variation, from just 2 types to 3, with the mixed type being the most common.
It is, and this calculation demonstrates the core principle behind the most important concept in population genetics, the Hardy-Weinberg equilibrium, HWE.
Okay, this is a big one. What does HWE state?
What Hardy and Weinberg independently figured out is this. In the absence of any evolutionary forces, no selection, no mutation, no migration, random mating, large population size, two things happen. First, the allele frequencies, $1 versus $2 in the population, do not change from generation to generation.
So P stays P, Q stays Q.
Right. And second, the genotype frequencies, A1, tolls, also stabilize after just one generation of random mating, and they reach a predictable ratio based on those allele frequencies.
And that predictable ratio is the famous formula.
If PUVs is the frequency of allele, $1, and a $2 is the frequency of allele, A101, and remember, up plus Q, Guelero, so is just one tuplet, then the expected genotype frequencies at equilibrium are P2 tolls for A10101M, 2P tolls for A101 toll, and 2 tiller 2s for A2
So $2 plus 2PQ plus Q2 equals A0. That describes the population structure when nothing is happening evolutionarily.
Precisely. It's the equilibrium state.
But why is this equilibrium state, this picture of no evolution, so incredibly important for studying evolution? You called it the null model.
Because science often progresses by testing against a baseline of no effect. HWE provides that baseline for evolution.
Yeah, so
Think about it. If you go out and measure the actual genotype frequencies in a real population, and you calculate the allele frequencies from that,
you can then calculate what the genotype frequencies should be if the population were in HWE.
exactly, using people2, 2pp2, and 2pp2, then you compare the expected frequencies under HWE to the observed frequencies you actually measured.
And if they don't match, if there's a statistically significant difference,
That's the signal, that's the red flag waving, saying something interesting is happening here. It tells you that one or more of the assumptions of HWE must be violated.
meaning evolution is occurring at that locus.
Yes. It means selection might be favoring one genotype, or maybe individuals aren't mating randomly, or maybe there's migration, or the population is small enough for random drift. HWE is the tool that lets us detect the footprint of evolution in real populations. It's absolutely fundamental.
Okay, so segregation shuffles alleles at a single locus. But organisms have thousands of genes. What shuffles combinations of genes at different loci?
That's where the second great shuffler comes in, recombination. This happens during meiosis, the process of making sperm and eggs.
When the paired chromosomes swap pieces.
Exactly. Homologous chromosomes, the one you got from your mom and the one from your dad for, say, chromosome 1, they physically line up next to each other and exchange segments. It's called crossing over.
So if you inherited an allele for tallness from your dad on chromosome 1, and then an allele for, I don't know, fast metabolism nearby on the same chromosome,
Recombination means those two alleles don't have to be passed on together as a fixed block. The chromosome can break between them, and maybe that tall allele gets combined with the allele for slow metabolism that you inherited from your mom on her copy of chromosome 1.
it breaks up linked combinations.
It constantly breaks up associations between alleles at different loci on the same chromosome. It generates new combinations of alleles.
And the key effect of this, according to the text, is that it combats something called linkage disequilibrium. That sounds complicated.
It does, but the concept is fairly straightforward once you unpack it. Linkage disequilibrium, often abbreviated as LD or just D, simply means that alleles at two different loci are found together in the population more or less often than you'd expect just by chance, based on their individual frequencies.
Okay, give me an example.
All right, let's use those hypothetical genes again.
Hmm.
Locus A with alleles A dollars A2, and locus B with alleles B dollars beta2. Let's say the frequency of $2 is 50%. And the frequency of B dollars is 5 and 4.55, and the frequency of BG2 is also 50%, PB2, 5 and R at 5.
Okay.
If the loci are independent in linkage equilibrium, the chance of finding an A$2 allele and a B$2 allele together on the same chromosome or gamete should just be the product of their frequencies. 0.5 times 0.5, or 0.25, or 25%.
Makes sense. Random association.
But if we look at the actual chromosomes in the population and find that the A2B2 combination occurs, say, 40% of the time, that's way more than the expected 25%.
Then they're statistically linked somehow.
Exactly. They are in linkage disequilibrium. The alleles 2R and 2L2 are associated more often than expected by chance. LD can also be negative, meaning they're found together less often than expected.
And recombination's job is?
Recombination's primary effect is to erode linkage disequilibrium. It breaks apart those non-random combinations, A-2 with B-2 in our example, and shuffles things up, constantly pushing the population back towards linkage equilibrium, where the association between alleles at different loci is random.
Let's try to visualize this with figure 4.9 from the text.
Yeah.
It shows 3 populations.
Right. 3 simple populations, each with just 8 gametes shown. And the key point is, all 3 populations have the exact same allele frequencies. In all 3, the frequency of $2 is 12, and the frequency of B$2 is 12.
So based purely on allele counts, the populations look identical.
But look at how those alleles are packaged together on the chromosomes. Population 1 shows maximum positive linkage disequilibrium. The value D is plus 14.
What does that mean visually?
It means the only combinations present are A1B11 and A2B2. They are perfectly coupled. If you have A$2, you always have B$2 with it in this population.
Okay, completely linked. What about the other extreme?
Population 2 shows maximum negative linkage disequilibrium, DB-14. Here, the only combinations are A1B2 and NW2LO1 lens. The alleles 2 and 2 are never found together. They repel each other, statistically speaking.
Got it. And population 3?
Population 3 is in linkage equilibrium, D0. Here, all 4 possible combinations, A1B11, A1B22, 2LBO, and A2B201 and A2B2 wives are present in equal frequency, 2 gametes of each type.
So, knowing the allele at locus A gives you no information about the allele at locus B. They're randomly associated.
Exactly, and this matters profoundly for evolution because, well, think about selection. Selection might strongly favor a particular combination of alleles, say TD2 with Vi with B$1.
If that combination gives the highest survival or reproduction,
then selection will act to increase the frequency of that specific A2B11 chromosome, thereby creating or increasing linkage disequilibrium. It's building that specific package because it works best.
so selection builds LD.
And recombination tears it down. It's this constant evolutionary tug of war. Selection tries to create optimal combinations of genes, and recombination tries to shuffle everything back towards randomness. The balance between these forces shapes the genetic architecture of populations.
Okay, if part 2 was all about shuffling the existing deck of genetic cards, this section, part 3, is about where brand new cards come from. Cards that have literally never existed before in the history of that lineage.
This is mutation, the ultimate engine of novelty.
The source.
Absolutely. We define mutation, quite simply, as any error that happens when DNA, or in some viruses RNA, is copied. Replication isn't perfect. Mistakes happen.
And while recombination is powerful for generating new combinations,
mutation is the only process that creates truly new alleles. It's the fundamental source of the raw genetic variation that allows populations to adapt to changing environments or exploit new opportunities over the long term. Without mutation, evolution would eventually grind to a halt once recombination had shuffled the existing alleles into every possible combination.
Right. So we can roughly categorize these errors, these mutations. We touched on point mutations earlier.
Yes, the small-scale changes affecting just a single base pair. As we said, they can be synonymous, silent, non-synonymous, changing an amino acid, or they might create a premature stop signal,
which usually wrecks the protein.
Often, yes. It truncates the protein, leaving it incomplete and usually non-functional.
And again, we have to remember those point mutations happening in the non-coding regions.
Critically important. A single base change in a regulatory sequence, that oven dial analogy, can dramatically alter when, where, or how much a gene is expressed. This can lead to huge changes in the organism's form or function, even if the protein sequence itself is untouched.
Okay, so those are the small tweaks. What about the big dramatic changes, the structural mutations?
Right. These are large-scale alterations involving chunks of chromosomes, sometimes thousands or even millions of base pairs. Figure 4.13 in the text illustrates the main types.
Let's go through them. First couple are kind of opposites. Deletions and insertions.
Yep. A deletion is just what it sounds like. A segment of the chromosome is lost. An insertion is the addition of a chunk of DNA, which might come from elsewhere in the genome or even from something like a virus.
And these can be really bad if they happen inside a gene, right? Because they mess up the reading frame.
Exactly. Since the genetic code is read in non-overlapping triplets, codons, deleting or inserting a number of bases that isn't a multiple of 3 shifts the entire reading frame downstream from that point. It's called a frame shift mutation.
So all the codons after the mutation are read incorrectly.
Totally garbled message. Usually leads to a completely non-functional protein very quickly. So deletions and insertions within coding regions are often highly deleterious.
Okay, what's next? The text highlights duplications as being particularly important.
Hugely important, yes. A duplication is when a segment of a chromosome gets copied, resulting in 2 or more copies of that region sitting side by side or sometimes elsewhere in the genome. It's genetic redundancy.
And you said evolution loves redundancy. Why again?
Because it provides the raw material for innovation. Think about it. If you only have one copy of a gene that performs an absolutely essential function.
Like breaking down sugar for energy.
Right. Any mutation that damages that single copy is likely to be harmful, maybe lethal. Selection will keep that gene sequence highly conserved, resisting change.
Okay. But if a duplication event happens?
Now you have 2 copies. The original copy can continue doing its essential job, remaining under strong purifying selection. But the second copy, it's now sort of expendable.
It's free to change without killing the organism.
Precisely. It can accumulate mutations without severe consequences. Maybe those mutations slightly alter its function. Maybe it becomes specialized for a slightly different sugar. Or maybe, over a long time, it accumulates enough changes to acquire a completely new function.
Ah, so that's how new genes arise.
It's one of the primary mechanisms. Many gene families, groups of related genes with distinct but related functions, likely arose through ancient duplication events followed by divergence. Think about the globin genes that make different oxygen-carrying proteins for embryos, fetuses, and adults. Or the huge number of genes involved in our immune system or sense of smell. Duplication provides the raw material, the genetic clay, for evolution to sculpt new functions.
It's not just a mistake. It's like a potential starting point for something new.
Absolutely. It's a key evolutionary enabling event. Okay, next structural change. Inversions.
Flipping a section around.
Yep. A segment of the chromosome breaks off, flips 180 degrees, and reattaches. The genes are all still there, just in reverse order within that segment.
Does that cause problems?
It can, but the main evolutionary significance of inversions often lies in their effect on recombination.
How so?
When a chromosome with an inversion tries to pair up with its normal, uninverted homologue during meiosis, they have to form these awkward loops to get the genes aligned correctly.
Right.
And crossing over, recombination within that looped region often leads to non-viable gametes, sperm, or eggs with missing or duplicated genes.
So the practical effect is?
It suppresses recombination within and near the inverted region. It essentially locks together the alleles inside that inversion, forcing them to be inherited as a single block, a super gene.
So inversions can maintain strong linkage disequilibrium even if recombination is happening elsewhere.
Exactly. It can preserve a set of co-adapted alleles that work well together, preventing recombination from breaking them up.
Interesting. Okay, 5th type, fusions.
Chromosome fusion is when 2 chromosomes that were originally separate stick together end-to-end, becoming one larger chromosome.
So that changes the total chromosome number.
Drastically. For example, humans have 46 chromosomes, 23 pairs. Our closest relatives, chimpanzees, gorillas, orangutans, have 48, 24 pairs.
So somewhere in our lineage.
There was a fusion event. It's thought that 2 ancestral ape chromosomes fused to form the large human chromosome 2.
Wow. And the text shows that chromosome numbers vary wildly across species, right? Figure 4.14.
Incredibly so. Fruit flies have 8, we have 46. Some deer species have only 6 or 7. And then you get things like ferns. Some species have over 1,200 chromosomes.
1,200. How does that even work?
It's complex, but these fusion events, and the opposite, fission, splitting, are major drivers of karyotype evolution, the evolution of the chromosome set, and can contribute to reproductive isolation between populations.
Okay, and finally, the most extreme mutation of all.
Whole genome duplication, WGD, also called polyploidy.
This isn't just duplicating a segment, it's duplicating the entire set of chromosomes.
Exactly. Usually it happens through an error in meiosis, where the chromosomes fail to separate properly, resulting in a gamete, an egg or sperm that is diploid, 2N, instead of the normal haploid N.
So it has the full double set.
Right. And if, by chance, two of these unreduced diploid gametes fuse during fertilization,
You get an offspring that is 4N instead of 2N.
you get a tetraploid, 4 copies of every chromosome.
That sounds like it would cause major problems.
It often does in animals, but it's surprisingly common and often very successful in plants.
Mm-hmm.
Many important crop species, like wheat, cotton, potatoes, are polyploids.
Why is it more successful in plants?
Plants seem generally more tolerant of changes in genome size and chromosome number. And a huge consequence of WGD is that it can cause instantaneous reproductive isolation.
Oh.
That new tetraploid 4N individual. If it tries to mate back with the original diploid 2N population, their offspring would be triploid 3N. And triploids are usually sterile because their chromosomes can't pair up properly during meiosis.
Ah, so the tetraploids can only reproduce successfully with other tetraploids.
Often, yes. Which means, in a single generation, the whole genome duplication event has created a new population that is reproductively isolated from its ancestors. It's basically created a new species overnight, genetically speaking.
A massive evolutionary jump.
Okay, so we've seen the types of mutations, from tiny point changes to whole genome duplications. But a key question is, how often do these things actually happen?
Right. We started by saying DNA replication is incredibly accurate.
It is. The cellular machinery for copying DNA and proofreading it is phenomenal. But phenomenal isn't perfect, especially when you're copying billions of letters.
So errors do slip through.
They do. The overall answer on mutation rates is they're generally low, but they vary hugely.
Depending on the organism,
Yeah.
the type of mutation.
All of the above. And even different regions within the same genome can have different mutation rates. Figure 4.15 in the text gives some sense of the variation.
Let's hit those numbers again, the precise ones versus the fast ones.
Okay, at the highly precise end, you have something like the bacterium E. coli. The error rate there is estimated at maybe 1 mistake for every 10 billion base pairs copied.
10 billion. That's incredibly accurate.
Astonishingly so. Humans are a bit less accurate, maybe around 1 mistake per 100 million base pairs copied during the formation of sperm or eggs.
Still very accurate, but noticeably higher than E. coli.
Yeah, about 100 times higher, roughly. But now compare that to the other end of the spectrum, RNA viruses.
Like flu HIV.
Exactly. These viruses use RNA as their genetic material, and the enzymes that copy RNA are much sloppier, generally lacking good proofreading mechanisms. Their mutation rates can be incredibly high, somewhere in the range of 1 in 1,000 to 1 in 100,000 bases copied.
That's millions of times higher than E. coli.
Thousands to millions, yes. It's a massive difference.
And that explains why they evolve so quickly, why we need new flu shots every year. They're constantly generating new variants.
Absolutely. That high mutation rate fuels their rapid evolution, allowing them to quickly adapt to host immune responses or antiviral drugs.
And you mentioned earlier, this high rate might even be adaptive for the virus.
It's thought so, yes. In the constant arms race between the virus and the host's immune system, being able to generate new variations rapidly is a huge advantage for the virus. So natural selection might actually favor viruses with sloppier replication, even though many individual mutations might be harmful to the virus itself. It's a trade-off.
Okay, so rates vary wildly. Now, let's talk about the effects of a mutation when it does happen. We need to introduce a concept called pleiotropy.
Right. Pleiotropy is a really important and maybe underappreciated concept. It simply means that a single gene or a single mutation in a gene often affects multiple, seemingly unrelated phenotypic traits.
So changing one thing has ripple effects.
Exactly. It's very rare for a gene to influence only one single characteristic of an organism. Think about that robot arm analogy again. A mutation affecting the gene controlling the robot's main motor speed. It doesn't just change the speed. Maybe the increased speed also causes more vibrations, affecting precision, uses more energy, affecting efficiency, and wears out the joints faster, affecting lifespan. One change, multiple downstream effects.
So a mutation that affects, say, bone growth might also affect calcium levels in the blood or kidney function.
Potentially, yes. These pleiotropic effects mean that genes are often involved in complex networks. When selection acts on one trait, it inadvertently pulls and pushes on other traits connected through pleiotropy. This creates genetic correlations and can constrain or channel the pathways evolution can take.
Makes sense. Okay, now the big question about effects. Fitness. Are mutations usually good, bad, or indifferent for the organism's survival and reproduction?
The short answer, based on many experiments like those shown in Figure 4.1 sedum, is that most mutations that actually have an effect on fitness are deleterious. They are harmful.
They reduce the organism's ability to survive or reproduce.
Correct. A smaller fraction are neutral, having no detectable effect on fitness. These might be synonymous mutations or changes in non-critical parts of non-coding DNA, and only a very tiny fraction are beneficial, actually increasing fitness.
And this goes back to the idea of tinkering with a complex machine.
Precisely. Most genes and proteins have been fine-tuned by millions of years of natural selection to do their jobs reasonably well in their current environment. A randomly changing an amino acid in an enzyme is far more likely to disrupt that finely tuned function than it is to improve it.
Throwing a wrench in the works is usually bad.
Usually bad, sometimes neutral. Very rarely, the wrench lands just right and makes things work slightly better.
The text mentions a human cost of mutation. What's that about?
It refers to the constant influx of these mostly deleterious mutations. Estimates suggest that, on average, each human baby inherits about 1 or 2 new deleterious mutations that were not present in either parent.
Wow, every generation.
Yep. Now, the saving grace is that most of these new deleterious mutations have only small effects, maybe reducing fitness by less than 1%, and crucially, they are often recessive.
Meaning you need to inherit 2 copies, one from each parent, for the bad effect to show up.
Exactly. In heterozygotes, who have 1 good copy and 1 bad copy, the harmful effect is often masked. This allows these deleterious alleles to persist at low frequencies in the population, hidden from selection most of the time, but they still represent a kind of genetic load or burden on the population.
Okay, so mostly bad, some neutral, but the diamonds in the rough are the beneficial ones.
They are the diamonds. Extremely rare, but absolutely essential. It's the gradual accumulation and spread of these rare beneficial mutations driven by natural selection that results in adaptation and the evolution of complexity. There is the fuel for all positive evolutionary change.
One last crucial distinction regarding effects. Where the mutation occurs matters hugely for inheritance. Germline versus somatic.
Yes, this is fundamental. For a mutation to be passed on to the next generation and potentially contribute to the evolution of the species, it must occur in the germline.
Those are the cells that produce the gametes, the eggs, and sperm.
Right. Only mutations in those cells will be incorporated into the offspring. Somatic mutations, on the other hand, occur in the regular body cells, skin, muscle, liver, brain, whatever.
They might affect the individual.
Oh, hugely. Somatic mutations accumulating over a lifetime are a major factor in aging and the primary cause of nearly all cancers. But
They die with the individual.
exactly, they're not passed on to the next generation. So, from an evolutionary perspective, somatic mutations are dead ends. Only germline mutations matter for the long-term fate of the species gene pool.
Right. We've established mutations happen. They vary in rate and effect. Most are bad, some are good, and they have to be in the germline to count for evolution. This leads to a really deep, almost philosophical question the text tackles. Is mutation random?
Ah, yes. And specifically, random with respect to what?
Right. The question isn't whether mutations happen predictably at certain DNA locations. Sometimes they do, hotspots.
Yeah.
The question is, does the need of the organism influence which mutations occur?
Exactly. This goes back to the pre-modern synthesis debates. The Lamarckian idea, in a nutshell, suggested directed change, that if an organism was exposed to, say, a poison, its body would somehow generate the right kind of mutations to confer resistance in response to that challenge.
So the environment would instruct the genome on how to mutate beneficially.
That was the idea. But the overwhelming conclusion from a century of genetics is no. Mutation is random with respect to fitness. Meaning the chance that a specific mutation will occur, say, one conferring antibiotic resistance, is not affected by whether or not that mutation would be helpful to the organism in its current environment. Mutations happen due to errors in replication or DNA damage, regardless of their consequences.
So the variation arises first, by chance.
And then the environment acts as the filter or selector. It favors the pre-existing variations that happen to be beneficial in that context, but it does not cause those beneficial variations to arise in the first place.
And the classic, definitive experiment proving this randomness is the Liederberg replica plating experiment. It's so elegant.
It really is a beautiful piece of experimental design by Esther and Joshua Liederberg. Let's walk through it using figure 4.1 as a guide.
Okay, step 1.
They took E. coli bacteria, which are normally sensitive to a virus called T1 phage, and spread them on a standard Petri dish with nutrient agar, the master plate. They let the bacteria grow up into numerous distinct colonies, each starting from a single cell.
And critically, the location of each colony on the plate is fixed.
Yes, their spatial arrangement is key. And there's no T1 phage on this master plate. It's a non-selective environment.
Okay, step 2.
Replica plating. They took a piece of sterile velvet stretched over a cylindrical block. They gently pressed the velvet onto the surface of the master plate.
Like an ink stamp.
Exactly. The velvet picks up a few cells from each colony, preserving their exact spatial pattern. Then they pressed this velvet stamp onto one or more new sterile Petri dishes. These are the replica plates.
So the replica plates have colonies in the exact same positions as the master plate.
Perfect copies of the colony layout. Now, step 3. The selection.
Introduce the virus.
Right. The replica plates contain the T1 phage, so almost all the bacteria on the replica plates were killed by the virus. But a few colonies survived. These were phage-resistant mutants.
And the Liederbergs noted the exact locations of these resistant survivor colonies on the replica plates.
Yes. Now, here's the crucial test. If resistance mutations arise in response to the phage, the directed mutation hypothesis, then the resistant cells should only be found on the replica plate where the phage is present. The corresponding colonies on the master plate, which never saw the phage, should still be sensitive.
But if resistance mutations arise randomly before exposure, the random mutation hypothesis,
then resistant cells should already exist in the colonies on the master plate at the corresponding locations, even though they were never exposed to the phage.
So what do? Step 4.
They went back to the original master plate. Using the locations of the survivor colonies they'd mapped on the replica plates, they took samples only from those specific corresponding colonies on the phage-free master plate.
So they picked cells that had never encountered the T1 virus, but whose sister cells on the replica plate had proven resistant.
Exactly. And then they tested these cells from the master plate for resistance to T1 phage.
And the result?
They found that the bacteria from those specific spots on the master plate were indeed already resistant to the phage.
Boom.
Proof.
Definitive proof.
The resistance mutations had occurred randomly, by chance, before any exposure to the selective agent, the phage. The phage didn't cause the mutation. It merely revealed which pre-existing colonies happened to carry it.
It confirms that variation is generated randomly, blindly. Selection is the non-random force that sorts through it.
Perfectly stated. It's a cornerstone experiment of modern evolutionary biology.
Okay, before we wrap up this chapter on variation, the text does briefly mention other ways traits can be passed down, ways that don't involve changes to the DNA sequence itself. Non-genetic inheritance.
Right. While standard evolution primarily focuses on genetic inheritance, it's worth acknowledging these other mechanisms exist, though their role in long-term evolution is debated. First, there's epigenetic inheritance.
We touched on epigenetics earlier, changes in gene expression.
Yes. These are chemical modifications to DNA, like methylation, or to the histone proteins that package DNA. These modifications can turn genes on or off without changing the A's, T's, C's, and G's. And sometimes these epigenetic marks can be passed down through cell division and occasionally even across 1 or 2 generations via sperm or eggs.
So an environmental effect on the parent could potentially alter gene expression in the offspring without a DNA mutation.
Potentially, yes, for a limited time. It's a hot area of research, but its long-term evolutionary impact compared to genetic change is still unclear.
Okay, second type.
Maternal effects. This is simpler. It's when the mother's phenotype, her physical condition, her environment, what she puts in the egg, directly influences the phenotype of her offspring, independent of the alleles the offspring inherits.
Like a well-nourished mother having larger babies.
Exactly. Or the amount of yolk in an egg determining hatchling size, or stress hormones from the mother affecting offspring development. These are real effects passed from one generation to the next, but they're not based on the offspring's DNA sequence changing.
Got it. And the third type, especially relevant for us.
Cultural inheritance. This is huge for humans, obviously, but also seen in some other animals. It's the transmission of information, behaviors, knowledge, skills, beliefs, traditions across generations through learning, teaching, and imitation, not through genes.
Language, tool use, social norms.
All examples. Cultural evolution can happen much, much faster than genetic evolution because it doesn't rely on rare mutations and slow changes in allele frequencies. It's a parallel and very powerful track of inheritance, especially in our species. Hashtag hack outro.
All right, that brings us to the end of our deep dive into Chapter 4 of Evolution, Mutation and Variation. We've really covered the bedrock concepts today.
We have. We started with the absolute necessity of variation as the fuel for selection. We looked at the genetic toolkit: DNA, genes, codons.
We saw how sexual reproduction through segregation and recombination constantly shuffles the existing deck of alleles, pushing populations towards Hardy-Weinberg equilibrium unless other forces are acting, and fighting against linkage disequilibrium.
And then we focused on mutation from tiny point mutations to massive structural changes like duplications and whole genome duplication, identifying it as the ultimate source of all new genetic novelty.
And critically, we confirmed through the Liederberg experiment that this crucial process, mutation, happens randomly. It's blind to whether its effects will be good, bad, or neutral for the organism.
The variation arises without direction. The environment only provides the test, the challenge.
So this really sets the stage perfectly for where we're going next in Unit 2. We've got the raw material, this randomly generated variation. Now what happens?
Now we bring in the engine. Next time, we turn to the genetical theory of natural selection. We'll explore how selection takes this raw, random variation and shapes it, non-randomly, driving the spread of beneficial alleles and leading to adaptation.
We start putting the math to selection, seeing how fitness differences play out over generations.
It's the transition from understanding where the possibilities come from, mutation and variation, to understanding the process that makes certain possibilities inevitable outcomes, selection.
Okay, but before we go, let me leave you with a final thought to chew on, something that ties back to the probabilities we talked about today.
Right.
We learned that beneficial mutations are really rare. And even when a truly advantageous one does pop up, initially, it exists as just a single copy in one individual.
Highly vulnerable.
And basic probability, just random chance in who reproduces and whose gametes happen to form the next generation, what we call genetic drift, means that even a beneficial mutation has a very high chance of just being lost by sheer bad luck in those first few generations before selection can really get a grip on it.
Yeah, the odds are stacked against a new mutation, even a good one when it's rare.
So considering that, considering how low the rate of beneficial mutations is, and how likely they are to be lost by chance when they first appear, how incredibly lucky, statistically speaking, were we? How lucky was life on Earth that the specific and probably long sequence of just the right beneficial mutations needed to build complex life, things like multicellularity, nervous systems, consciousness, the human brain actually managed to survive that initial lottery of chance and spread successfully through populations over billions of years?
It's a question that definitely highlights the interplay of chance and necessity in the evolutionary story. A lot of improbable steps had to happen.
Something to ponder. Thanks for joining us for this deep dive into Bookfulness. Join us next time as we get into the mechanics of natural selection itself.
Looking forward to it. Until then, keep digging into the details.